Coming up on Mayo Clinic Q&A. While there are effective injectable vaccines to fight COVID-19, drug makers are creating vaccines that will be easier to store, transport, and administer. Currently, Mayo Clinic is developing a new nasal vaccine platform that may have advantages in blocking the coronavirus.
The goal is to make protein from the coronavirus to have the immune system see it, and then if you actually got infected, to reject the virus.
Welcome everyone to Mayo Clinic Q&A. I'm Dr. Helena Gazelka. Thanks for being here today. The COVID-19 pandemic has brought public awareness to vaccines and how vaccines work in a way that we've never seen before. A vaccine is an agent that causes the immune system to remember a specific disease-causing entity. In the case of COVID-19, that's a virus, of course. And then when the body sees that entity again, or that virus, it remembers it and helps to prevent future infection. At Mayo Clinic, decades of research have led to the development of a new vaccine platform, a single-cycle adenovirus nasal vaccine. This is now being tested in phase 1 clinical trials for COVID-19. Well, I'm excited today to have Dr. Michael Berry with us. Dr. Berry is the director of Mayo Clinic's Vector and Vaccine Engineering Laboratory, and he's going to explain to us about these vaccines today. Thanks for being here today.
Thank you. It's great to be with you, and I'll do my best to try to explain and translate from scientists to everyone else.
Well, I think that's fantastic, Mike, because I have about 10 questions just after reading the intro that I could ask you about. First of all, starting with how to say Vector and Vaccine Engineering Laboratory quickly.
Well, it's easier if you say it slowly. So.
That's right.
But,
well, tell us about this, Mike. What is a single cycle adenovirus vaccine?
Yeah, well, so maybe I'll take a step back and kind of talk. You gave a great background on what vaccines are. For, to take a step back from that, most of what we are is made up of proteins, and the same thing goes for the virus. So actually, when most of the vaccines that are being deployed, and many of you have gotten, are actually, the goal is to make protein from the coronavirus to have the immune system see it, and then if you actually got infected, to reject the virus.
That's really interesting, Mike. So the thought is that, or the concept is that our body should know what belongs to us. And when it sees things that don't belong to us, it will recognize it as something that's bad. So having more of that protein isn't a bad thing, in that it makes the body recognize it even better and mount a better response. Would that be fair to say?
Yeah, so in theory, and at least in practice, when we test these, if we put equal amounts of our vaccine versus like a regular adenovirus vaccine, that same amount, ours will drive a much stronger immune response, and so the level of protection is higher. The other potentially cool thing is that if ours is really 100 times more potent, then maybe if we make a batch of an adenovirus vaccine, we could make a batch of the ones that are out there, or the batch of ours, we might be able to squeeze out 100 times more doses out of the same batch. So, that could be very important on a global scale, for COVID or for other sorts of pathogens.
That's a wonderful point. What does it mean that it's single cycle, Mike?
Well, it's sort of a vector, jargon sort of thing, but it basically has to do with that multiplication. So when the adenovirus puts the gene in the cell, the ones like the Johnson & Johnson or Oxford one, that one gene, that $1 bill goes in, and it just remains in that state, and it makes a lot of the spike protein, but ours will go in and it will copy that gene many times, so that the more copies of the gene, more protein you produce off of it. So the single cycle means it's basically going through part of its cycle and making a lot more protein.
Okay. And Mike, what is the difference between a platform and a vaccine, or are they the same thing?
Well, so you could call, if it was, you made a vaccine and it was sort of what one might call a one-off, of you made it once and it only applies to that pathogen. A platform means that you can kind of plug and play or cut and paste. So before we did the SARS virus, we actually did a bunch of other things like HIV, influenza, Zika, Ebola, Clostridium difficile. So, and it's pretty simple because you just basically cut and paste the gene for the protein you want to target into the virus, and then you can target different pathogens.
That's really interesting. So you can, well, this has to be specific for the virus or the pathogen that you're trying to fight. You can make this specific for different uses.
Right. Yeah. Yeah. And
Wow.
so over the year, you know, when I was a baby scientist about almost 30 years ago, we used things called gene guns, you know, so it was literally something you would shoot the genes in, and that, you know, and you could plug and play whatever you wanted with that, but it was not very efficient. And so we evolved over, you know, the past few decades, moving towards these adenoviruses because they're very robust. Evolution's done all the heavy lifting for us to be efficient in getting genes in there. But also, you know, as you mentioned, one of the sort of the strategies is to actually try to vaccinate not in your muscle, but intranasally,
Yes.
because, you know, and almost never does a virus enter your body in your muscle. And so one would say that maybe that's not the place you should put your vaccines. And it ends up that your body is well evolved to try to repel things that come up your nose or in your eye or at what are called mucosal surfaces. So it's engineered all these cells and antibodies to be up at that mucosal barrier to try to protect you. And so there's data even with this our vaccine that, you know, there is an advantage of actually vaccinating not in the muscle but intranasally, and particularly when you're talking about a respiratory virus like SARS, that's actually has a pretty good strategy.
Isn't that interesting, Mike? I had literally never thought of that. I think of things, you know, I'm a pain physician, so I think of things in terms of how does the body absorb them or whatever, or how can you make it bioavailable to you like a drug. But with a vaccine, maybe it's more targeting the area where the virus or the pathogen would live. Or,
yeah, and it's like, you know, and it sort of comes down to sort of trafficking, you know, like if you grew up in your neighborhood, you're going to tend to come back to your neighborhood. The same thing goes with your immune system. If you learned in your nose or your lungs, you're probably going to come back there more often. And so therefore, if you get exposed to a respiratory pathogen, the likelihood of you seeing it and doing something about it's much higher if you're constantly driving by the old neighborhood.
Mike, one of the things that has been astounding, I think, to anyone who works in a science or any research area is how quickly science has changed over the course of COVID-19 and how fast information has been shared, et cetera. When you talk about this vaccine platform, does that mean, and about plug and playing for different pathogens in the future, does that mean it will be much faster even to develop vaccines in the future as we see new pathogens?
Yeah, and I think we're seeing that with the messenger RNA vaccines and to some degree the adenovirus vaccines. The fastest vaccines you can make are DNA vaccines, and every other vaccine technology, including ours, at some point starts off with a DNA. And so if you want to make messenger RNA vaccine, you first make the DNA and then you use it to make the messenger RNA. And then we also use DNA to make our virus. So the fastest ones can be DNA and messenger RNA. The viral ones are a little slower because you have to make the viral vector, but the payoff is they're a lot more potent generally when you kind of run them head to head against each other. So I think the response rate is really fast, and technologically, the slower part is the regulatory process of, you know, so how long, you know, you have to do the clinical testing to make sure things are safe, and I think some of the groups that went forward, like Moderna and some of the other ones, had some advantages because they'd already worked on SARS-1, you know, the original SARS virus. So they could kind of use that to give them a foundation to jumpstart quickly.
Say, Mike, we have talked quite a lot about COVID vaccines in the past and how those who are immune compromised may not respond as robustly to them. And there are some vaccines, I think, that are not necessarily safe in those who are immune compromised, perhaps with live pathogens, et cetera. Is there any reason that this sort of a platform would work better for those who are immune compromised?
Yeah, I think they will, just because they're more potent. It's kind of pound for pound. But we haven't tested that in, like, particularly. I think it'll be interesting to see how well they do in older people. As you get older, your immune system becomes a little bit less robust, and it may take a harder poke to activate it. And so you kind of see that with the current flu vaccines, that they tend to give higher doses, more protein in older patients to try to get the immune system up. So the immune compromised people, it would sort of depend on exactly how they were compromised. But our vaccine and the others are engineered to not, cause problems. So I think they should be more potent.
But are intended to cause a more intense response of the immune system, hopefully overcoming that. If there was just a minimal response, you may not be covered against the pathogen.
Yeah, and I mean, the other thing we've tried to do is kind of make it hard on ourselves, is to try to see how good we could do if we just immunized once. And that's kind of a measure of potency of, if you have to come in and boost with your vaccine, then you're probably not as potent as one that can come in once. And so we just are completing a study in experiments where we immunized once and then challenged with the SARS virus 10.5 months after that one immunization. And, we saw good protection, after that very long period of one immunization. So yeah.
And besides the fact that the immunization is more robust, or the response is more robust, I think of the global improvements in being able to administer vaccines if you only had to reach each individual in the world once.
Yeah, and actually that's a whole other thing that we could talk about, of like some of the things that we work on is, how can you make these more easily to deliver. And so at certain times we've done things like take the vaccine and put it into capsules so you could just swallow it and then be immunized. So that the idea would be you could potentially ship the vaccine off to other countries, and it should be pretty stable at normal room temperature. But then rather than have needles and syringes all over the place,
Right.
a pill or something you put under your tongue, could be quite good.
Not to mention the freezers that have been medical grade freezers that have been needed for some of the vaccines that we're using now.
Yeah, and I mean, that's the adenovirus vaccines have actually, I think something on the order of 500,000 people, particularly military people, have gotten adenovirus vaccines that were freeze dried and then given as a pill.
Interesting.
To protect against adenovirus itself. So there's a
Wow,
lot of
that is really
higher
interesting.
part.
So we talked about this being in phase 1 trials. What does that mean to our listeners? How close is this to being available for use in humans?
Technology was licensed to a company and they are taking it into humans. And so the first thing you have to do is make sure it's safe. So this is the first time the single cycle platform's been tested. So the phase 1 is the first test. And, with the pandemic, and all the, already pretty great vaccines that we have, how many far along we get with the testing in the phase 1, 2, and beyond, it's hard to say. But now with the new variants popping up, it's kind of persisting the pandemic, I think. And so we may be able to get pretty far along with testing it. But that's sort of up to the company that's licensed it.
Wow, that's great. And Mike, we talk so much about how in the silver lining of COVID has been this rapid dissemination of research and information, but actually these vaccines, not just the one that you and I are talking about today, but the ones that are in emergency use authorization now were decades in the making. Would that be correct? How long does it take to work on this, and how long have you been working on this project?
Well, like I said, I think 28 years ago, I was shooting vaccines with gene guns. And so you could say it's been 28 years. This particular platform has evolved. I think we've been working on it about 13 years. And this was work done by, really talented graduate students and technicians at Mayo Clinic. So it's these things may benefit people, but it's also like we're also educating and building up the people for the next generation of, okay, what's coming next. And so if there are people out there who have interest in pursuing higher degrees like PhDs or MDs or master's program degrees in the area of gene therapy or vaccines or oncolytic viruses, Mayo is a great place to come. So I would encourage you to check it out.
Well, that's wonderful. One of our three shields: patient
Yeah.
care, education, and research, and all so important to have combined together. Say, Mike, I have a question for you. We've heard so much about vaccine hesitancy. So people saying, oh, they just cranked this vaccine out too quickly. We don't know if it's safe. What would you say to that? Is it really, have the vaccines that we are seeing in use now for COVID-19 really been put forth that quickly, that they haven't been tested properly? Would you have concerns about that?
No, and I've been vaccinated, so I think the cost benefit is the huge of I don't want to end up in the hospital with SARS, so I definitely think it's worth it to do. I mean, as I said, I've been doing this stuff for almost 30 years, and so these vaccines some of them have been around even longer than that. So, and they have been tested in a number of different diseases like HIV, or like in 2014 when the Ebola sort of potential pandemic was percolating in Africa. One of the two vaccines that went into human testing was an adenovirus vaccine. And so these have been around. It's just, we've never immunized so many people with them. And
Yes.
like I mentioned, the about 500,000 people have gotten the military vaccine. It was an oral vaccine, so it's a little different. I think there may be, like we were talking about the intranasal and whatnot. I mean, your body's engineered to deal with viruses at respiratory surfaces. And so it may be that, those types of routes of oral or intranasal may have some safety advantages as well, but we won't know until we test.
So basically, we could say they've been decades in the making, these vaccines.
Yes, exactly.
Mike, I find it absolutely fascinating, this conversation with you. And one of the thoughts that I have had, because you know that I've done some work with Greg Poland, Dr. Poland, who also has worked in vaccine research, et cetera, and does at Mayo Clinic. But, vaccines are something that we take for granted. I have adult children now, I have grandchildren. We just expect that kids get vaccinated and that we don't have some of the horrible diseases that plagued our forefathers and the generations that came before us. What is it like as a vaccine researcher to finally see people excited about vaccines and talking about vaccines?
Well, it's great. I mean, sort of like you say, if vaccines work, they're kind of boring, I mean, so it's kind of very interesting that, like I teach in our virology and gene therapy courses. And so it's like now the vaccines have become a big chunk of those lectures. So that's very exciting. And, the vaccine hesitancy is, I think it's a matter of what information people are consuming. And, and Dr. Poland's a great communicator on this topic. And many of us scientists are not that good at talking to the lay public, because we sort of exist in the state of we have facts now, and then we're kind of wishy-washy. We almost never like to say always or those types of things. And that's, doesn't help us, telling people. If you talk to, all the science or most scientists will have been vaccinated, and there's a good reason for that.
Mm-hmm. Well, I'm just delighted to get to talk to you because I loved what you said, that vaccines can be kind of boring, but people have no idea the work that goes in, the decades and decades of research that goes into seeing the success in the end. And so how wonderful to be able to laud that a little bit today.
Yeah, it's very exciting. And, actually, I've been at Mayo 15 years, and actually Dr. Poland recruited me, because I try to make vaccines, and he does a lot of the testing actually in patients. And so, there's a nice bridge there. So it's just
wonderful.
Been nice to not have a pandemic drive it all.
That's right. We can do without that, can't we? But it certainly has gotten you working hard over the time, I'm sure.
Oh, yeah. Yeah. So
any last words you'd like to share with us? What else would you like our listeners to know today, Mike?
Well, it'd be great if everyone would get vaccinated. And, I think it's, being vaccinated is the best way to be protected. And, I don't think any of us want to end up in the hospital with this disease. And so it's just a good way to go.
I couldn't think of a better ending myself. Thank you so much for being here today, Mike.
Thank you.
Our thanks to Dr. Michael Berry, Director of Mayo Clinic's Vector and Vaccine Engineering Laboratory. Thank goodness there are people like Mike doing the work that they do each and every day. We're so grateful for that. We thank you, our listeners, as well, for being here today. I hope that you learned something. I know that I did, and we wish each of you a wonderful day.
Mayo Clinic Q&A is a production of the Mayo Clinic News Network and is available wherever you get and subscribe to your favorite podcasts. To see a list of all Mayo Clinic podcasts, visit newsnetwork.mayoclinic.org. Then click on Podcasts. Thanks for listening, and be well. We hope you'll offer a review of this and other episodes when the option is available. Comments and questions can also be sent to Mayo Clinic News Network at mayo.edu.